Hello. Good morning. Good evening. Good afternoon. Good night. Welcome to 2700, where we're going to be talking today about the Lophophorata. The Lophophorata. Where we left off last time was talking about one of the enigmatic kind of mysteries, remaining mysteries of animal systematics, animal phylogenetic systematics, and that largely has to do with the placement of these taxa, the Bryozoa, the Brachiopods, and the Phorinids. Now you can see I've put the Tribbiani kind of quotes around it because as it is portrayed in your Dunn phylogeny, is this a monophyletic group? The answer is no. Right as it is portrayed right here. And you can see that quote at the bottom. The Lophophorates have had a long and torturous taxonomic history. This is at least the second or third time that we've heard the word torturous applied to the taxonomic history of one of our lineages. So I think it's probably from now on just a good a priori decision to say, when considering the taxonomic history of a group, presume it's torturous. Where were we? Well, we were talking about the Gnathopherans with the gnathing jaws of the rotifers and the Chaetognaths. And we talked about how the placement of the Chaetognaths was resolved with phylogenetic systematics using deeply conserved regulatory gene regions called Hox genes. And it consolidated the otherwise formerly outside of the Spiralians, the Chaetognaths, into the group with the rotifers and two other phyla, which we'll get to, these parasitic ones, but largely described by the actions of their jaws, morphologically, and lots of genetic support. And then we burrowed into the Lophophorata, this coelom, this kind of ring-shaped mouth, in a otherwise bilaterally symmetrical organism, this apparent radial symmetry associated with cilia running around tentacles that in cross-section have coelom extending into them. And we had talked about, with reference to the rotifers, the importance of this hydrostatic skeleton, these antagonistic muscles firing, longitudinal and circular muscles firing against the hydrostat. I made the comparison that the hydrostat of the hydrostatic skeleton is kind of like a water balloon, and those muscles are your hands squeezing the balloon around it or along the side of it. And we left off with the consequences of the phylogenetic placement, and we're going to come to that in a second. But one of the things, I guess just these are the outcomes of the thought processes that I'd like you to go through with these narratives, these taxonomic narratives for these Lophophorata phyla, is to think about, appraise, synthesize, and interpret the evolution of the Lophophore, and think about different larval strategies, and then interpret and synthesize examples of similarity and difference across these three taxa, both extant and through the fossil record. We'll talk about that as we go. The first of the three, so these are the Phorinids, the little muppet, a hand puppet, and then we've got Brachiopods here, and this is a tiny Bryozoan where you can see that ciliary action kind of like a buzzsaw on the outside of that Lophophore tentacle. So there's the characteristics from chapter 17 in the grouping of chapter 17 that talks about the Bryozoans. There's about 5,000 extant species, many, many more in the past, and we'll talk about that. Some people call them moss animals because that's actually the derivation of the word. Bry means moss, and animal means zoa, so moss animals. They're largely colonial. That's what you're looking at here, is this arrangement of these individual little animals into a colony. Largely hermaphroditic, although there are some that are gonochoric, and they live both freshwater systems, but historically are most diverse and abundant in the marine system, in the saltwater system. Here's a little bit of juxtaposition. Look on the left-hand side with these blue. These are the traits. Remember, this is a way of showing traits, common or absent. So common in this case is the extension of the blue line. So Bryozoans here, and the red tips are just like phyla where we have been already or where we are going. So we've got The Bryozoans possessing a brain, possessing nerves, possessing a basement membrane. So that epithelial membrane really being a characteristic of the bilaterian of metazoan animals outside of these very, very comparatively simple ones. They have a through gut. There is no transient anus here like the ctenophores. There is a full-on anus. There is a mesoderm, so a central body cavity, epidermis, endodermis, mesoderm, and a coelom, and a a blood vascular system. Now, those are the kind of array of the characteristics. With this side of these kind of two contrasting phylogenetic hypotheses, with the red dots, those kind of stamps from the bar where they are, this is the arrangement of the either monophyletic here with the Lophophorata being supported as a clade, a monophyletic clade within the spiralia, or here where it is outside of, so the Brachiopods and the Phorinids being a group, a monophyletic group, but the Bryozoans representing another lineage. Now the Bryozoans are fantastically diverse in the fossil record. What do you mean, Smith? Well, take a look at this. There's two large lineages. That's the two different colors. So they got the stenonomates and the gymnomates. You don't have to remember those words. If I wanted to, I would explain what they were if this was ever on a quiz. But what you've got here is time, shown in two kind of axes on the left and right, with the era, the geological eras, Paleozoic, Mesozoic, and Cenozoic, and then we've got millions of years ago. So deep time down here. And then the width of these shapes, that kind of, all of those different shapes, indicates the ebbs and flow of diversity of the number of genera, okay, the amount of diversity. And so here's a little scale bar for that. And what I want you to notice, don't focus, there's say three different colors there, but take a look at this line here at the end of the Permian, where there was, for most extant lineages, and there was a range, a whole bunch of different diverse groups, most ended at the Permian extinction. One extended through, began to diversify, and then suffered another great extinction at the end of the Mesozoic. And then one small lineage prior to the Mesozoic expanded and became all of the species of moss animals that we have today. So when we're talking about the extant lineages, I want you to think about the extant lineages being a subset of all of the things. And where can you see all of these other things that didn't make it through the Permian, for example? Well, you can see them, these moss animals, in the rock structures that are in and around the Guelph area and in and around other bits of limestone that are arrayed around the different parts of Ontario. So there's my swamp thing that makes me think of moss animals. These are not swamp things. These are my parents. These are my lovely parents. And they had gone out with that broom into the fields beside our place in December of 2020, and they took I shipped a Gigapan to them. They took the Gigapan, this like panoramic camera robot that you ran into probably in 1070 in the dairy bush, and instead of pointing it out into the landscape, they pointed it down at the rock face. And what they did then was record the fossils, outcrops that were in and around that area. So they captured some of the, in this case, Ordovician diversity that included things like crinoids and cephalopods and gastropods and brachiopods and bryozoans. So let's take a look at one of those potential groups here and bring this over and zoom in. So you see this little bit of kind of reticulate, that is the colonial structure, the fossilized evidence of a colonial organism, the bryozoan, that existed 450 to 500 million years ago on the bottom of a tropical sea that is now the Ottawa Valley. There's another crinoid that's over here, and you can kind of, if you scan this QR code, you can go to a whole list of these things that we've arrayed for you for your enjoyment if you're not close to rocky outcrops of limestone wherever you sit. Now, that colonial structure in the Ordovician limestone that my big panoramic picture that my parents took 5 years ago, is also apparent. These are scanning electron micrographs of extant living bryozoans today. So these are all called zoocea. These are multiple. So zoocea, individual zooceum. That's the name for the space, the house that the individual bryozoan lives in. Zoocea means literally animal house, which makes me think that some good homework, if you haven't seen it already, is go and watch the incredible Belushi in Animal House. So here's a cross-section from your text, chapter 17, where we can see the black line there that's tracing up to the shape, the exterior part of the zooceum. And we've got connections through the septum with the pores that connect in through the funiculus, this base, into each of the individual kind of bryozoan zooids. And then outside of the zooceum can extend the feeding tentacles, and the feeding tentacles of the bryozoan are the lophophore, right? That's why we include that. So inside of this we have the coelom, and then we have, and that's mostly the mesosome, so that's most of this space, the metasome, and the zooceum, extending out into the tentacles and the lophophore. And here's another look at a stained example, a stained bryozoan, because it shows kind of nicely the U-shaped gut. And the colony is comprised of many, which is one of the synapomorphies. So one of the many discrete casings that they secrete, these tubes or shells, are derived from chitin that they produce, so protein, or calcium carbonate that they derive from their surrounding waters. And also, they've been described as having a long and tortuous taxonomic history. Here's both more simple, but also a little bit more complex. Lots of different lines going in from the older text that we used to use in this course. What I'd like you to see here is some common elements, points of connection between individual bryozoan zooids. One slice through the lophophore, through an extended lophophore, one where the lophophore is drawn in, so we've got a retreated, protected from maybe a grazing predator here, and then one that's extended and feeding. And you can see this gray that the coelom is extending out into that tentacle. So there we've got the mesocele out in the tentacle, and we've got the metacele in terms of the body, and we've got lots of different muscles shown in these long lines that extend down into the body, and of course the zooceum that is the hard casing, the animal house. So they have an introvert, which is the anterior part of the body where the lophophore and tentacles can be withdrawn. You kind of saw that right here, where this is the lophophore that is extended in the drawing on the right. The funicular system is what connects the individual zooids. Ciliated cells, of course, ciliated being the action of cilia being a lot of the action of the lophophore that creates feeding currents. And then they have, the arrow here is showing you a coelomopore, which is this ciliated groove right here where eggs are released into the environment. So the musculature of these animals, here's the first, some other kind of morphological characteristics. The first one is the funiculus. So a ligament or a tendon that connects the stomach to the bottom of the body wall to keep the organism in place, protected in its zooceum. There's some staining that shows you like what one would actually look like here. Feeding, not feeding, retreating from predators are again accomplished with antagonistic muscles. So we have circular muscles and longitudinal muscles, but we also have, and we've got another, in this case, the circular we'll call, or in this groups are called parietal muscles, and then a longitudinal muscle that is at the base of the zooceum is called the retractor muscle. So increasing the parietal, kind of like the circular muscle, squeezes the water balloon so that the lophophore is extended. And that's what's happening in A, B, and C. Now, and that everts the lophophore and allows for feeding. Now, squeezing the here we've got the retractor muscles. When the parietal muscles are relaxed and the retractor muscle fires, that pulls the lophophore back into the zooxea and protects it. Now I mentioned, and you can kind of see it here a little bit in the diagram, the U-shaped gut. That's what we're looking at right there. And it is a through gut. Now, if you were to follow that line up, you can see, so the mouth, such as it is in the middle of the lophophore. And thankfully, when pooping, the bryozoan does not poop. So there's the mouth right there. And what you're going to see is the video of this bryozoan pooping. You'll see where the anus exits, which is thankfully outside the lophophore, right there. So physiologically and physically integrated zooids. So they share coelom, they share coelom, they share all the metabolic byproducts of what they're feeding. So this is a true colony. Now, how they live, we've used that historically. We have some terms. Just remember, we had the different kind of terms to describe the complexity and the shape of different sponges. There are some similar terms for bryozoans, and I'll show you them in a second. But more analogous to like things like other colonial animals, like social animals, where different ants do different jobs, different zooids, different individual bryozoan bryozoas perform different jobs here. And you're looking at SEM here down that jaw-like mechanism. That's called the avacularia. That's used for defense to little nip away little small predators. They have a vibracularia that is probably used for cleaning the surface of the colony. It's the Roomba. And so there's the avacularia, here's the vibracularia. Now the autozooids, these are the classic ones that we've seen already. That's kind of what I would imitate with my hand, kind of extending out of the zooxeum of the sleeve of my jacket. So lots of polymorphism. Now that's the polymorphism of the zooids, of the individual kind of bryozoan animal. But how the colony, it can be stolonate, so it looks almost like a coral. It can look like a leaf. It can look more like a lettuce. But these are all different shapes. And again, not so much phylogenetic groupings, but just a description of the form that we perceive in the water. Now, the most common form is an encrusting form, where on the leaf of kelp or something, you would see these kind of tiny pixelate shapes from which extending these gorgeous little individual zooids. These are autozooids that are all out there feeding right now, and here's a still image. And the idea in some cases with the encrusting form is that the thinking is that is providing some protection, because it's very hard to get for an animal. It's easier for an animal to nibble off of the fructose or stolonate form, but on an encrusting form, it's harder to get their mouth around it. But of course, there are many, many creatures that can do it. And so here, what you're going to see moving around. So there's an encrusting form on some kelp. And the chitinous exoskeleton is good against many smaller grazers, but there are big things like this fish that can go in. So that lichen-looking shape on the leaf, that is the colony of bryozoan. And so while the individual zooid may derive some protection from this, and if there is a specialized predator, a grazer in this case like this, it can peel off whole chunks of it. Now, the colony is, as we've said, physiologically and physically integrated. There's been some neat studies that have been done in the literature, but also by fields, undergraduate students on things like the Marine and Freshwater Field Course that we offer through our department, where in collecting bryozoans, they will apply a passive dye. A dye, water, yeast and water, but that has in this case, the Congo red dye. And what you find is the distribution of that dye tracks which of the zooids are sharing, which are feeding, and which are not feeding. And so you can see in this close-up the areas that have no dye. These are non-feeding zooids that are likely used for reproduction, and then the distribution of the dye showing you these individual kind of pixels or zooids that are feeding zooids. And there's a close-up here. So these are looking at, we've broken up, here's the exit for the zoocoeum, and we're zooming in to these little plate-like connections that show kind of gorgeously the intimacy of connection in between. So there at 300 micrometers is the scale bar, and if we zoom into these little connections, these would be the actual connection points, the pores in between donor and receiving zooids. So there's the diagram kind of flipped here with the two connections, and there's the connection plate through which communication, direct connection and sharing of the coelom would happen. Now, some of those non-feeding zooids that you saw, those are producing all sorts of different kind of larvae and releasing them out into the water. So they have larval types, an array of different larval types. You can see many of them do have ciliary bands. So the adults are characterized by cilia, so are the larvae. Some of them look even mostly like cilia. Now, this is a Siphonauts larva here. And the Siphonauts is a long lived, like it can live for months and disperse quite a far distance. So it's a planktotrophic versus a lectotrophic. These three other larval types are lectotrophics. So in the red, lectotrophic are things that are, they're bequeathed a yolk. So they feed. They have something as they emerge and are released into the water column. They don't need to feed themselves. They are given that traveling lunch right away. The planktotrophic, the blue, the Siphonauts, it travels and needs to feed and live in the water as a smaller planktonic organism as it's moving around. So there is a period of settling and metamorphosis, but I wanted to show you this Siphonauts, kind of the little astronaut that's going away and permitting potentially escape from decaying or colonizing new areas because it's living out in the plankton for a long period of time. It really does look like that last drawing. So the Siphonaut larva there. Now these organisms can also reproduce asexually. So each colony can arise from an asexual budding or an ancestrula that's formed by a sexually produced larva or from a statoblast. And there's a statoblast right there and there. And they can also travel long distances as they bud off and form great densities in the water when that period of release is happening. Bryozoans and the symbiotic organisms that live within them, we're actually very dependent upon them. So I wanted to throw this bit in as a bit of pharmaceutical eye candy for you to say, who cares? If you're saying, who cares? If you maybe live with people who are like, oh my God, invertebrates, who cares? Bryozoan, Lophophora, what? Well, bryozoans are the source of one of the most useful compounds that has been used in the eradication of HIV/AIDS. It's been used in cancer immunotherapy, in the treatment of Alzheimer's disease. Bryostatin is a compound that's only found up until 2018 from one species of marine bryozoan, Bugula narratinia. And we harvest it aggressively for this compound. It takes 14 tons of the bryozoan. 14 tons to make 18 grams. So a yield vanishingly small that was in some articles that I was reading was characterized as saying to make a salt shaker of this compound, you need 3 elephants worth of the material. And that's prohibitive. It costs about $1.3 million on the open market for a single gram of this material. And it's not actually the bryozoan that is the source of the drug. It's a symbiotic bacteria that's found only within the species of bryozoan that makes the bryostatin. And in the wild, we think the compound protects it's another element of protection for the larva, in this case, from predators. Fish are repelled by it, for instance. And perhaps into the future, we'd be able to synthesize it without harvesting tons and tons of bryozoans. So that's one of the Lophophorate taxa, the one that sits outside, where its phylogenetic position is it the Lophophorata, as is shown here, not monophyletic, or is it the Lophophorata, a monophyletic group? So this is the Phoriniid worms. Worms, not a worm. A worm is an annelid, a segmented thing. This is worm-like. Not a super diverse group right now, but they have some amazing adaptations that we're going to talk about. Now, sometimes with the derivation of the name of the phylum, we can give you some element of their biology. Moss-like looking moss animal, Bryozoa. In this case, there's no real help. Phoronis was apparently a mystical priestess of Argos who was loved by Zeus. That doesn't help me. I love to think of they look like a Muppet. They look like that looks like the most beautiful little sock puppet. This is a specific kind of actinotroch larva that the Phoronids use. So we talked about Siphonotes larva as one of several different larval stages. So we're going to compare and contrast some larval stages here across these Lophophorate groups. The Phoronid we have a bulbous end that almost acts like a root. These generally sit deep in anoxic soil. Then they have a trunk that extends out through another kind of a body casing. And then the lophophore extends out. And it can be quite whorled and complicated. We'll take a look at that. So they're marine, and they live in these, and they like gorgeous, right? Like this, amazing. Looks like the evolutionary endpoint of the I think it was formerly known as ribbon gymnastics. Like, the lophophore is the only thing that extends above. And it's really tight and solid molecular evidence that groups them very closely with the brachiopods. So of the three Lophophorate bearing taxa, it's really the Bryozoans that we're uncertain about. The brachiopods and the Phoronids, that's a good group. A large drop in diversity compared to like the hundreds or thousands historically of species that we're talking about with the Bryozoans. Not that many species. It's more than 14. But it's less than 100, say, extant ones. And just because it's another reminder for me to say they're called worms, they're not worms. That worms as a taxonomic phrase really, we should replace it with vermiform. Vermiform means worm-like. So worm-like. And even in this case, they look less worm-like to me than they do almost like, I don't know, cnidarian-like. They're marine. They're sessile. They don't move. They're benthic. They're in the benthos. They're in the substrate. They're externally bilaterally symmetrical with an internal asymmetry and a left side dominance. And that starts to set up the whorl inside. Now the whorl is really kind of cool. It brings us back to some of the things we were talking about as we entered the metazoa, as we left the protozoa behind and we entered the metazoa. We started talking about real limitations or guidelines that are applied to evolution by physics to say like surface area to volume ratio in this case. And so what we see here is a real expression of that when you're very small, you have no need for a huge increase in the surface area of your lophophore, this feeding and respiratory part of your body. But as you get bigger, you need more and more complex spirals to provide enough surface area to feed the increase in volume that your body is going through. It's cool and beautiful. Now, one of the synapomorphies for this group is that they have corpuscular hemoglobin. Now, you think this is not a big organism, millimeters, centimeters long, and it's not moving or it's sessile. Why is there all of a sudden the appearance of an oxygen-carrying hemoglobin in their respiratory, in their circulatory system? This generally, when we see the addition of these oxygen-carrying molecules, this is about a thing that is doing a job and is moving around, and or it's big and it's complex, and we need to carry the oxygen from the outside of the body to the inside of the body where lots of work is being done by muscles. So why is diffusion not enough for this? Is it I've phrased it as a question, but likely this is an adaptation to living in anoxic or hypoxic conditions. So the lophophore extends out of the mud and into the water. mildly oxygenated water, but the water is not circulated to the rest of the tube, to the rest of the organism, the rest of the body. So diffusion wouldn't work very well when the majority of your body's sitting in a tube where there's not a lot of water movement at all. Now, a couple of things that probably you're familiar with from biochemistry, but if not, just a short kind of thinking about hemoglobin. When hemoglobin is not contained inside of a cell, when it's extracellular, it's big. When it's intracellular, it's small. And it's equal kind of concentrations of hemoglobin compared between extra and intracellular. Small ones can bind more oxygen since there's a greater surface area. But that can change the osmotic pressure, meaning that it would be harder to release at the site where it's needed, where the work is being done. So small hemoglobin can bind more oxygen, and making it cellular changes that osmotic pressure and allows you to release it rapidly where it's needed. So the Phorinids have come up through evolutionary time with this amazing packaging of small hemoglobins intracellularly, and therefore enhancing their oxygen transport while minimizing the effect on the osmolarity of the blood and allowing it to be released and do its work where it's needed, which seems to be kind of a fantastic adaptation, a physiological, biochemical adaptation to life in an anoxic place where a lot of your body is held captive within a tube inside of the deeply anoxic material. So oxygen as a bridge to oxygen transport in invertebrates, which with an aside saying we'll talk a lot more about this if you take 3700 next year. Nemertine worms, annelids, mollusks, phoronids, echirids, another kind of annelid and echinoderms can have transportation molecules, but mostly it's extracellular. We've come to a point where I'm going to ask you to pause me in a second, okay? And I'll say that there are nucleated red blood cells circulating in body fluids in 5 invertebrate phyla: echinoderms, mollusks, annelids, phoronids, and nemerteans. Okay? What I'd like you to do is pause me in a second and find each of those phyla on the tips of this Dunn phylogeny. Keep me paused as you answer the question, this trait, nucleated red blood cells. When did it arise? Did it arise only once? Has it been lost many times? Has it never been lost at all? Those 3 questions. Answer them for yourself. Annotate it. Answer the questions. And then start me up again. And I'll give myself a pause with a number. And I'll take a drink. And we're back. So there you go. So brachiopods don't have it, right? So that's a big chunking and there is variation within each of the phyla. The phyla is not a uniform distribution. So within annelids, 5 marine polychaetes have it. What's a polychaete? We'll talk about that at the end of the week. 2 large molluscan classes have this. Nemertean worms have it. Brachiopods don't. And then half of the orders of echinoderms do. So what's the question? Did it arise one time back here and then was lost on many different occasions? Or did it arrive 2 times and was lost within each one of those? So once within the deuterostomes, once within the protostomes, and then subsequent loss in annelid lineages and within all of the brachiopods. What's the most parsimonious assumption there? Well, if we're going to presume one time, that's one time is parsimonious. However, to explain the extant distribution of the trait involves a lot of losses. That makes the evolution one time less parsimonious than saying probably 2 times, probably at least 2 times. We're going to unpack that further with subsequent phylogenies, but just another experiment in plotting traits on trees to think about evolutionary processes. Back to the Phoronids in their chitinous tubes. That's not a secret chitinous tube, which would be kind of more fun. The Phoronids in their secret chitinous tubes. They secrete the chitinous tubes, and they incorporate sediment particles into the tube. They never, this makes, actually I'm going to link to a secret chitinous tube that they never leave. They secrete the chitinous tube, they never leave it. It has it's closed at the bottom. The anterior part is where the lophophore is, but you can see when I'm talking about the distribution of oxygen, this is where the water is, up here with the lophophore. The rest of the body can extend quite deeply. The coelom can create the hydrostatic skeleton, and then the parietal, similar kind of setup of muscles that the bryozoans have to either squeeze circularly on the coelom and fire the lophophore out, or to retract and bring it back in. The lophophore, with the last bullet's kind of cool, is that it can be it can be grazed on. They can regrow it. Now, onto the Muppets. This is the hand puppet. This is the actinotroph, or the actinotroph larva. That's free living. It's free living. It's part of the plankton. Another current that runs through the river of this course, the thread in the sweater that we are building and wearing in our exuberant evangelizing about invertebrates, is the fact that if you're sessile as an adult, likely you have a larval stage that moves, that can colonize new areas, that can potentially be invoked as an escape for a destruction or a degradation of habitat. So, the siphonaut larva for the bryozoans, the actinotroph larva for the phoronids. So both of the adults are part of the sessile benthos. The larval stage is part of the plankton and is moving around and is part of that food web, the same kind of food web that we talked about with the ketognaths the other day. And that, again, if you want, come back, and in 3700 we'll spend several different lectures talking about plankton. They have a loss of chaetae here on the growing lophophore compared to brachiopods. File away the brachiopod, we'll get there in a second. Now, one of the cool things, so we've got here a comparison of actinotroph larva forming or in a cross section, and the siphonaut larva. So both of the planktonic forms of the two lineages we've seen so far. And then, don't look at me, but look at the gorgeous video of this little thing. So we've got this, take a look at the actinotroph, so we've got that. And you can see the hindgut and a comparable hindgut in the siphonaut larva. And lots of cilia and lots of feeding going on through the mouth, the pre-oral hood, or the front part, the tip of the sock. Now, very quickly, and that's the approximately 30 minutes there, when the siphonaut or the actinotroph fall out of the plankton onto the substrate, there's an enormously rapid developmental change that happens where that gut changes from a linear gut to the U-shaped gut that characterizes this group. They start to secrete a tube, they connect themselves to the bottom, and then, ta-da, you have a benthic adult. The last of the groups today is the brachiopods. This is the arm-footed things. Brach, the arm, pod, foot. Now this is an amazing video from a remotely operated vehicle that was operating off the coast of Alaska a couple of years ago that I've clipped because it shows you an open brachiopod. Now, if you haven't been exposed to brachiopods, you might be going, Smith, that's clearly a clam. It's like, no, this is the first and most successful of the bivalves. And we'll talk about whether it's a bivalve or a bivalve in a second. But what I'd like you to notice is zoom, lean in and take a look. Oh, of course, the camera zooms back out. Stupid ROV. That I control. And you can see branches of the lophophore and a ciliated edge here on the edge of the mantle that's kind of making the shell. So brachiopods here, much more diverse than extant species than the phoronids, and one of the most diverse lineages of the Paleozoic. A lot of the species that you see in the Silurian limestone here in Guelph, or in the Ordovician limestone in different part of the Devonian limestone of different parts of Ontario where it's exposed, are these apparently clam-like looking things that are not mollusks at they are these brachiopods. Lots of shared parts. So the U-shaped gut, in this case, an absent anus. And these are the two shells, valves. The important thing here is that one of the valves is dorsal and one is ventral, as opposed to the clam where the shells are lateral. So here, dorsal and ventral. Gonochoric stick, a rudimentary circulatory system, solitary, compare and contrast with the Bryozoans, benthic, like all three lineages we talked about, and marine. Pause as the recording to cough. One of the cool things about, so I mentioned they're called arm-footed or the brachiopods. That's me with the arm-footed. But they're also called, in some communities, called lamp shells because they resemble ancient Roman lamps. There's the brachiopod here, and here's an ancient Roman lamp. So if you're familiar with ancient Roman lamps, bully for you, this is a lamp shell. So one of the synapomorphies for these group is the presence, of course, of this lophophore, this crown circled in red here that you could see in the last diagram or the last kind of video from the remotely operated vehicle off the shoreline of Alaska. Hollow, coelom-filled tentacles encircling a mouth. There's gas exchange and feeding. So really like mollusk. This is what the mollusk thing kind of copies, right? Convergently, they look really similar. Now, they're sessile or sedentary, and they use no distinct head here. And they have on the outside these chetae rim that kind of go all the way around the outside edge of the valve or the shell. They have this pedicel or pedicle, and that's the that which connects it to the substrate. And in some taxa, that's the part they eat. All right, so here's a really old remotely operated vehicle that is actually it's kind of using a propulsion system that lets it bounce across the lens, the benthos, and there's really nothing to say other than a really weird way of moving. But as it zooms in and out, this is off the coast, one of the coasts of New Zealand, and it's just to show that in the areas where there are still extant brachiopods, they can be quite dense. The last better video was more solitary, but they can be quite dense. So not many species today, and only a superficial similarity to bivalves. These are not looking the same because of shared evolutionary history. And in fact, let's dive into that even more and take a look at here on the left-hand side. We have lateral valves, a left and a right. That's the clam, that's making the pearl, that's the oyster. Okay, that's the left-hand side. And on the other side here, we have the brachiopod, where I've flipped it, so don't worry about the words. It's just to show you that instead of being here, we have here. So a dorsal valve and a ventral valve. So the plane of symmetry for the valve. The plane of symmetry in a brachiopod runs perpendicular to the hinge, and in the valves it runs right along the hinge. Now here we've returned to the Echinos dive here, and we can zoom in just a bit. And you can see the lophophore. You can see the ciliated tentacles. And then if you're really careful, you can see these are the chetae that line the edge of the mantle. And you can see them spread across, collecting little bits of detritus there, probably stirred up by the ROV. And this is another thread, right? Another thread, a common one, throughout the length of the courses. We historically could not see the living, we could not collect, we could not watch the living brachiopod. We dredged them, we dragged them with a bucket up from the bottom in the sludge. And so there's a bias there to hard-shelled things, away from soft-bodied things like cnidarians and ctenophores certainly, or even some bryozoans, but also away from rocky outcrops, because you can imagine dragging that shovel head across a rocky outcrop is going to miss. That was a bias towards muddy areas, sludgy areas. And now we fly these remotely operated vehicles, and we can collect and we can actually see. And I know that's a bias. Using that as a synonym to I understand is a bias of our species to be visual, but in this case it really lets us see them for the first time. Now, just like the brachiopods, their diversity, so it's different kind of a presentation, but different, whole bunch of lineages. Don't worry about the names, but look at the change. So up and down, periods of lots, high diversity, decline, high diversity, decline, but then all the lineages decline 251 million years ago. That's the end Permian mass extinction. Then the diversity, the rainbow of colors here, most of these lineages are absent from the 3 or 4 that survived and extended and diversified. Some of those, like Lingula, is an extant lineage that dates back to the Ordovician and is still here today. That's kind of amazing. What happened at the Permian? Why when they returned, why when the conditions changed after the extinction event did not all of the lineages survive? Well, just like we saw, so here's that brachiopod example, here's the Bryozoan example from the start of today's time. The end Permian was incredibly catastrophic. 250 million years ago, maybe 95% of the species on the planet did not survive. A marine fauna that's demonstrated from this article in Tree that pre-Permian would have had, say, 100 different taxa from many different phyla living on it was reduced to 4 or 5 individual species. When I was in this course, we were taught that this was largely due to volcanism. Volcanic change and volcanic winter could be one small element, but there's increasing research that thinks it's changes in ocean currents that changed coincident with rapidly rising temperature, creating enormously oxygen-free parts of the ocean. So, and acidification. So high temperatures, anoxic conditions, and acidification. Terrifying numbers. In an extent way, these are kind of 3 of the verbs. These are 3 of the things we're doing to today's ocean. Not good. So, taking a look at some of the last couple of seconds here, thinking about the dorsal and ventral, and we've got the Chidae here, and then we've got the Lophophore with some calcareous elements, like, so in this case, imagine the Lophophore, because you're inside a shell, right? And it has to open to feed, and that's where some of these muscles come in. And so the muscles are not retracting the Lophophore out like the Phoronid or the Bryozoan, but they're instead opening the valves to permit the Lophophore to be exposed to currents, feeding and respiratory currents. The pedicel is the attachment. And then the calcareous elements allow the Lophophore to be kind of hanging, not just limp, but extended out so that the surface area that it is exposed to the water is higher. So there we've got adductor, abductor muscles that are contracting to close or open the shell, and the pedicel that connects it to the substrate. There's the dorsal and the ventral valve. Here's taking a look at we've got some of the muscles. We've got a heart that is dorsal. This is strangely flipped because in this diagram, unlike the last, the ventral is on top, so I apologize for that. There we've got an adductor muscle that's going to fire to close the shell, and then the diductor muscle here that's going to fire to open the shell and expose, and then the pedicel that connects it. There's another, so we'll simplify the drawing. Here we've got the adductor contracted, shell's closed, adductor relaxed, adductor is relaxed, and the diductors are contracted and the shell is open, and that lets the Lophophore feed. The pedicel, in this case, is something that is consumed in some cultures. So that's an edible part of the brachiopod. And there, with the valve spread open, you can see the, what would that shape be? Almost like a figure 8 or an infinity sign of the Lophophore inside, protected inside of the valve. So dorsal and ventral valves, not lateral like in the mollusk, a large Lophophore, and the shell is secreted by a mantle edge, which is an outgrowth of the body wall. And they're sessile, They're benthic. All three of the lineages we've talked about today are. But they can move a little bit. They can move their pedicel to scissor the shell back and forth to create a right angle opportunity to the water current. If there's an evident water current, they will set themselves up to maximally expose themselves to that current so that the lophophore can feed and respire as efficiently as possible. These are the small bristles or chetae that extend from the groove at the edge of the mantle, at the edge of the valve. And they're like a tactile sensory thing. And the groove that many of the fossil shells show suggests that they also had this. These chetae are not homologous with the chetae we'll talk about on Friday with the annelids. Now, the lophophore extension, bunch of different, you've seen this drawing, you've seen this drawing. Now we're looking at the lophophore, these extensions kind of that let the lophophore sit almost like railings, like curtains hanging from a railing up into the water. Now that's creating circulatory systems. So they're exposed to the water, but also then upstream collection systems that then allow, so there's the brachium or the arms of the curtain rod, the curtains in this case are the lophophore. And then we've got an inhalant canal and then an exhalant canal. So the water comes in on one side of the curtain and out on the other. And then we've got food that's collected by the ciliary action of these little tentacles, dropped down into this brachial groove, and then sent in to the mouth. And so the water flow bringing both oxygen and food into the body. There we've got sexual, this is one of the sexual lophophorites, and they're larval, long-lived, and again, part of the plankton. So that's sessile adult planktonic larval stage, and kind of gorgeous looking. And there, so we've got an articulate larva and a inarticulate larva, and the inarticulate is what we're looking at here. So the tentacles extending out, very juvenile kind of rudimentary shell, and then settling, and less of a massive change that happens with the bryozoans and the Phorinids, because this larval stage, even though it's free living and free swimming, it has the rudimentary parts. You can kind of see where the adult brachiopod is going to come from there. It looks like it's the kids look like their parents. So this is our tour through the lophophorites, the bryozoans, the Phorinids, the brachiopods, comparing and contrasting larval stages or larval strategies where the adult is sessile, how do they feed, common elements, the lophophore, thinking about phylogenetic placement, thinking about ecology, and thinking about sensitivity to similar catastrophic events in the past, like the Permian extinction. That's it for today. We're going to talk more on Friday about annelids and other incredible invertebrates. So take care of yourself. Take care of those around you, and we'll talk soon.